This is the Professor, CBMD PhD, for today Saturday, December 14th, 2024. It is 9:40 and 58 seconds, p.m. Eastern Standard Time, and today's research, endosomal compartments. So let's hop to it here. The contents of vessels incorporated into early endosomal compartments remain in this compartment. As the early endosome matures, it becomes more acidic and sinks into deeper regions of the cytoplasm to become late sorting endosomes or late endosomes. The ESCRT cytoplasmic protein complex controls membrane remodeling, which is the basis of the sorting and maturation of early endosomes into late endosomes. The sorting function continues in late endosomes. The endosomal membrane of some late endosomes undergoes inward invagination to generate intraluminal vessels. These structures called intracellular multivesicular motifs or multivesicular endosomes are also sorted. Some mature into lysosomes where their contents will be degraded, and others fuse with the plasma membrane to release intraluminal vessels by exocytosis. These extracellular vessels originating from multivesicular motifs are called exosomes. Endosomes can be viewed either as stable cytoplasmic organelles or as temporary structures formed by endocytosis. Recent experimental observations of endocytic pathways conducted in vitro and in vivo suggest two different models that explain the origin and formation of the endosomal compartments in the cell. The stable compartment model describes early and late endosomes as stable cellular organelles that maintain their connection to the external environment of the cell and Golgi apparatus by vascular transport. Coated vessels formed at the plasma membrane fuse only with early endosomes because they express specific surface receptors. These receptors remain part of the early endosomal membrane. The maturation model suggests that early endosomals are formed de novo from endocytic vessels originating from the plasma membrane. Therefore, the composition of the early endosomal membrane changes progressively as some components are recycled between the cell surface and the Golgi apparatus. This maturation process leads to the formation of late endosomes and subsequently lysosomes. As this compartment matures, specific receptors present on early sorting endosomes, e.g. for coated vessels, are removed by recycling, degradation, or inactivation. The major function of early endosomes is to sort and recycle proteins internalized by endocytic pathways. As their name implies, early endosomes sort proteins that have been internalized by endocytic processes. The sorting process is primarily regulated by ESCRT. ESCRT represents an assembly of four separate protein subcomplexes, ESCRT 0 through 3, that mediate the scission of membrane necks from the inside. The shape and geometry of the tubules and vessels emerging from the early endosome create an environment where localized changes in pH constitute the basis of the endosome sorting mechanism. This mechanism includes the dissociation of ligands from the receptor proteins. In addition, the narrow diameter of the tubules and vessels may also aid in sorting large molecules, which can be mechanically prevented from entering specific sorting compartments. After sorting, most proteins are rapidly recycled or destined for digestion, and the excess membrane is returned to the plasma membrane. The fate of the internalized ligand receptor complex depends on the sorting and recycling ability of the early endosome. And this is Professor Christopher, codename Will, leader of research scientist ufologist, computer technician, computer programmer, philanthropist, writer, mathematician, artist, photographer, film producer, chef, animal, and nature lover, passion, world peace. And this video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.